So, how does eukaryotic DNA replication occur? First thing to note is there are differences between prokaryotic and eukaryotic DNA. Therefore, there are differences in prokaryotic and eukaryotic DNA replication, with eukaryotic DNA replication being a more complex process. Okay? And if you think about the different things you know, the size, the shape, the number, the compaction, all of these things play a role in DNA replication. So consider this. Average size human chromosome, 150 million nucleotide pairs. Single replication fork, 50 nucleotide pairs per second. 50 nucleotides per second for 150 million nucleotide pairs. How long will replication take? However, DNA replication occurs when? What is the name of that phase? I'm hoping that S phase came to mind. In mammalian cells, S phase is only about 8 hours. So how can we replicate 6 feet of DNA in 8 hours when we know it would take much, much longer than that? Okay, so the first thing is they have multiple origins of replication. So we have our linear chromosome, and as compared to bacteria where you have one origin and then you go out, we have multiple origins on the same chromosome and then multiple chromosomes replicating at the same time. Okay, so the first way to speed up this process, multiple origins of chromosome, more than one chromosome replicating at the same time. Now, for initiation, it varies. So we know in bacteria they have DNA, A, sites, and proteins. We know in yeast they have ARS elements. So these elements are about 50 base pairs. They are AT rich, similar that we've seen in bacteria. They have a consensus sequence, and then they have additional elements. So these are all at the origin and allow replication to take place in yeast. It's not so simple in other organisms. So in some, we don't know the DNA sequence. Like we looked at, or maybe there is no DNA sequence. So while ARS elements are in yeast and D and A are in bacteria, some of them don't have a consensus sequence. But now, wherever this origin is, the first thing is there's a pre-replication complex that must form. So here we have the origin recognition complex, or ORC, which acts as the first initiator of our complex. So the ORC comes and binds. After the ORC comes and binds, it recruits or promotes the binding of CDT1, CDC6, and then MCM helicase, which is a multi-protein subunit. Okay, so that has about 6 proteins in a group to form the MCM helicase. Then, together now, we have the initiation of our pre-replication complex. Now, in order to go from pre-replication to active replication, we're going to start to phosphorylate. So that means adding a P to some of our to some proteins in this area. When we phosphorylate, it's going to cause this CDT1 to come off, this CDC6 to come off, and this ORC to come off, leaving just the MCM helicase. And we know the job of helicase is to start to open up our DNA. Okay, so at the origin, you have to form a complex. These proteins come in, they form the complex. Phosphorylation occurs, these proteins leave. MCM is there to start to open up. And now it's going to proceed similar to bacteria. So helicase is unwinding, topoisomerase is there to relieve supercoils. Single-stranded binding proteins are keeping from the double helix from reforming. Now, it's interesting to note here, a difference is polymerase. So we know that there are 2 different polymerases being used. First thing is the RNA primers made by primase, which in this case is DNA polymerase alpha. So DNA polymerase alpha has primase activity. Then DNA polymerase epsilon is on our leading strand, synthesizing it, and DNA polymerase delta is on the lagging strand. Okay, these are incorrectly notated. There is a sliding clamp here pCNA, which is going to help this to be a processive enzyme, like we also seen in bacteria. And now this is going to go along with the leading strand continuously replicating towards the fork, the lagging strand discontinuously replicated away from the fork, and this process will continue. And there's Okazaki fragments on this lagging strand as well. So a lot of similarities and some major differences. Another difference is how RNA primers are removed. So if you remember in bacteria, DNA polymerase I removes the primers. Here we have something called flap endonuclease. So here we have Okazaki fragments and primers. And as we're going along, DNA polymerase gets to the Okazaki fragment and continues a little bit, causing the primer to flap out. These flaps are recognized by flap endonuclease. It's an endonuclease, which means it's removing things in the middle. So flap endonuclease comes along and removes the flap. Okay. And then we can continue replicating and creating a flap until it's completely gone. And so we'll continue this process until we remove the entire primer. Once the primer is removed, there's still a gap between one Okazaki fragment and another, and this is where DNase ligase comes in and seals that backbone. So polymerase can lay all the nucleotides, but it can't connect that phosphodiester in the sugar phosphate backbone, so DNA ligase does its job. It's also important to note here that if a flap is too long, so let's say this Okazaki fragment continued and this flap grew out to be too long, flap endonuclease can't remove the flap. Now we're going to rely on DNA2 nuclease / helicase to cut that flap so that it is a size that flap endonuclease can come in. So if a flap is too long, DNA2 nuclease / helicase will minimize the length of the flap so that flap endonuclease can remove the flap. But ultimately, flap endonuclease is chopping away at that primer flap by flap. All right, another difference we have is telomeres. So at the end of linear chromosomes, we have these repetitive sequences called telomeres. They have a consensus sequence, so to speak, so I'm giving air quotes to that. So if we look here, we see some of the consensus sequences, similar but different. These tend to be, they are 6 base pairs in length, and they are repeated, and they have a lot of guanine, okay? And let me note too, 6+. So most have 6, some have 6, some you'll see with more than 6. All have an abundance of G's, okay? So telomeres, short sequences, characteristic by having a lot of G's in it. So the reason we have these is to solve the end replication problem. So remember we talked about on the lagging strand, DNA polymerase can't link the first 2 nucleotides together without a primer. Well, we physically can't put a primer in here to allow it to replicate. So therefore, we would be left with areas that don't have any DNA. So with each time we replicate, this will get shorter and shorter because we just can't physically fit to replicate this. So to circumvent this problem, telomeres were created. And we know that telomeres are created on the DNA by telomerase. So telomerase comes in and it has a portion of it that will recognize the end of a chromosome. Then it's going to sit there and use itself because it has polymerase activity and it's actually reverse transcribing. So it's going to use the RNA it has to create the DNA necessary for the ends of these telomeres. As stated, this is turned off in somatic cells, okay? So with each time a somatic cell divides, the telomeres are getting shorter and shorter. Once the telomeres reach a critical length, 2 things can happen. 1, they apoptose. What does that mean? The cell dies. 2, they enter senescence, which means the cell is no longer dividing, but it's still present. Okay? So telomeres solve the end replication problem. But telomerase is turned off in somatic cells. So once somatic cells divide, the telomeres shrink with each division. If the telomeres reach a critical length, the cell can either die or stop dividing. Okay.